Hi, in this second video clip, we'll talk about our second cytoskeletal protein, microtubules. And as you can see, these are organized primarily radially from a central organizing structure called the centrosome. Microtubules are the stiffest of the cytoskeletal elements, and in contrast to intermediate filaments, which are sort of assembled, again, these add dimers of tubulin proteins. There's an alpha and beta subunit, and they are synthesized constantly, extending the microtubule or having it recede. Because they all start growing from the centrosome, they have a directionality that these are the plus ends of the microtubule and these are considered the minus ends of the microtubule. And finally, as their organization shows, they extend to all reaches of cells and serve as essentially highways for materials to travel along if they need to be transported to various locations within a cell. So here's the basic unit of tubulin. It's a heterodimer of an alpha and a beta subunit. These alpha and beta subunits are organized into protofilaments, of which there are 13 within a single microtubule. You can see an electron micrograph here where you can even make them out. And as we mentioned before, the polarity is very important. You have plus ends and minus ends of microtubules with the beta subunit, alpha subunit, beta subunit. So the beta subunit is the end subunit on the plus end. The lateral interactions between the protofilaments are non-covalent, which I think is a little bit surprising given the stiffness of the tubules themselves. In addition to the centrosome, which predominates during interphase, there are other organizing structures which microtubules can grow out of. During mitosis, there are two spindle poles that help drive migration of chromosomes to the center. When they line up, they will help participate in removing the chromosomes from their portion of the chromosomes from the center. They will help position the nuclei at large distances between the two dividing cells, and also can help generate polarity, as in the case here in the epithelial cell. This nucleus is not in the center of the cell. It's been pushed essentially by the microtubule network a farther distance from the apical end than the basal side, where the microtubule structures are shorter. Basal bodies are also the organizing structure for microtubules that extended into cilia. These microtubules interacting with each other can cause the wave-like motion of the cilia projected into the space, for instance, perhaps the digestive tract or respiratory tract, where it can function to help move things out of your lungs as part of a clearing process. This is a blow-up schematic of a centrosome. Inside the centrosome are two centrioles, which are themselves made up of microtubules. They'll play important roles when we get to cell division. In this schematic, we see that all the microtubules growing out of the centrosome have plus ends that are projecting. The minus ends are connected to the centrosome. It's a protein called gamma tubulin, which forms the connection between the centrosome and the microtubule projections. And as we see on the next slide, these projections can be regulated independently. They can be shrunk shorter, they can be regulated to be grown longer, and this can be done in a number of ways. But one of the ways of regulating is simply the amount of free tubulin present in the cytoplasm. If there are local concentrations with high amounts of monomers and the proper amount of ATP, those microtubules can tend to grow. The length of microtubules can influence the length of cilia, and there are a number of drugs that act by interfering with microtubules. This is one way of preventing cell division, since microtubule function is essential for mitosis. So this slide depicts the two processes of a growing microtubule and a shrinking microtubule. I think I said ATP earlier. GTP is the molecule that's important, and if there are a lot of free monomers and an ample supply of GTP, this will tend to promote growth. As the subunits have become incorporated over time, they will hydrolyze the GTP. So now GTP is indicated by red dots, but as time proceeds, the GTP hydrolyzes to GTP, and now we see only the green dots. If microtubules stall in terms of their growth, if there are not a lot of monomers available or not a lot of GTP, the hydrolysis process will continue. The formation of GDP inside the subunits favors the dissolution or the dismemberment of the tubulin subunits, and so this will start to shrink. It's not going to stay steady. It's going to tend not to stay steady at a certain length. Again, it's sort of a dynamic process. And here we simply see an electron micrograph which really supports this drawing of the peeling away of tubulin subunits from a shrinking structure. Because these microtubules are constantly growing and shrinking, on any average time approximately 50% of the tubulin subunits are in the monomeric form and the others are in the polymeric microtubule form. This slide is depicting the way in which microtubules can influence the shape of the cells and in particular the creation of cell polarity, which is really referring to an asymmetry here, as specialized sides. By stabilizing the growth of microtubules in one direction with these capping proteins, this will tend to keep the nucleus projected at a far distance from this. And for instance, this is a mechanism observed in epithelial cells where the nucleus is always closer to the basal rather than the apical layer. Now, as we've mentioned many times, the microtubules are sort of the highways or the transport paths of the cell. In this particular diagram, we see a nerve cell with a cell body. Here's where proteins are made. Here's where neurotransmitters are synthesized, and to get to the end of a nerve, which again, this can be a meter long, the nerves that innervate your large toes. So, but with a very narrow diameter, if you were going to rely on diffusion, it would probably be many years before molecules in any appreciable concentration at the cell body would reach the nerve synapse. But with active transport at a rate of about 10 centimeters per day, it can still take several days for synthesized molecules to reach axons. The vesicles moving away from the nucleus and towards the nucleus are interacting with the polarity of the microtubules. Remember the plus end and the minus end. And as we'll see on the next slide, different motor proteins move in different directions on microtubules. So kinesins are the motor proteins. These kinesins bear some structural similarity to myosin, and as you can see, the kinesins have sort of two different subunits that are part of the motor head that interact with the microtubules, and it's speculated that it's a sort of walking mechanism where one of these motor proteins will be anchored to the microtubule, and this leg, if you will swing forward, attach, establish itself. This attachment will come free. It will move ahead, and it's very much like a walking mechanism. Dyneins are the related proteins like kinesins, but they are the ones that move towards the minus end. Various kinesins have different shapes that will bind to cargo, which will specify whether cargo is supposed to be moved anterograde or retrograde. And even the kinesin proteins themselves, when they all get to the plus end, are shuttled back by dyneins to the center of the cell body. Microtubules are involved with the bending of flagella used for propulsion of cells by having motor proteins cross-link between the microtubules. The microtubules can be moved relative to one another in experimental situations where you don't have microtubules fixed. The microtubules will be passed along one another, but when they have cross-linking proteins which anchor them and limit the amount that they can be moved, the starting to translate one of these microtubules relative to the other, since they're linked, will instead cause bending. And this is the way, for instance, sperm are able to swim in solution. Kinesin motion is studied in experimental setups where single beads are attached to single kinesin molecules. This is done by a dilution study. And you can then, with time photography, measure the rates of transport, and with optical trapping where lasers can produce a force sort of essentially grabbing onto a silica bead, the forces can be measured that are generated by the two motor heads walking along a microtubule. And on this final slide we see sort of a walking mechanism. Each of these release and attachment cycles are powered by the hydrolysis of ATP. Although this slide doesn't indicate the foot over foot type motion, which most people suspect about these two motorhead movements. And I'm not expert enough to know whether this is true or not. However, I've seen enough of these videos where, as you can see, sometimes the white foot is in the lead, sometimes the black foot is in the lead. And this, I think, represents the consensus view as to how kinesins or dyneins move on microtubules. Okay, thanks for listening.